Imagine for a second using an AI, but like not just to draft a polite email to your boss or to write a really simple Python script to sort your desktop folders.
Right, something a bit more advanced than that.
Yeah, way more advanced. I want you to imagine an AI autonomously discovering a new drug or independently processing raw, completely uncharted genomic data to find, a hidden disease marker.
I mean, it sounds like absolute science fiction. But the reality is we are inching incredibly close to that exact threshold. The software is just getting that good.
It really is.
So the question we're facing right now isn't really if AI can do these things. It's more about how we can actually prove it mathematically and scientifically.
Exactly. And that is exactly what researchers at Agentic and Roche have just put to the test. They've built this thing called CompBioBench.
Yes, it's quite the project.
It is a massive, highly rigorous benchmark of 100 completely diverse computational biology tasks. And it's designed to test the absolute limits of general-purpose AI agents.
And our mission for this deep dive today is to really explore how you measure an AI's ability to do real messy science. We want to see what actually happens when we, take the training wheels off and just let these digital agents loose on complex, real-world biological data.
Okay, so let's unpack this because testing an AI in biology is uniquely difficult, especially when you compare it to testing it in standard computer science or math.
Oh, it really is. And to understand why CompBioBench is such a huge breakthrough, we first have to look at why existing AI tests just kind of fail to capture the reality of actual scientific work.
Right.
If you look at standard AI benchmarks, they mostly test math or basic programming. And those fields, they have systematic verification. It's binary, The code compiles and runs, or it crashes and spits out an error.
The math equation balances, or it just doesn't. It's a closed loop.
Precisely. But biological data, it is inherently noisy. It is underdetermined.
Wait. Underdetermined. Let's break that down for a second. That basically means you could have multiple biological theories that all perfectly fit the exact same set of experimental data, right?
Exactly. The data doesn't just speak for itself. It requires interpretation, context, and a really deep understanding of biological mechanisms to actually make sense of it.
I always like to use this analogy when thinking about this. Standard coding benchmarks are kind of like building IKEA furniture.
Oh, that's a good one.
Yeah, you are given the exact parts in a box, a super clear step-by-step schematic, and you just follow the instructions. You know when you're done because you're looking at a bookshelf.
Right, it's very prescriptive.
But computational biology is like being dropped into a dense forest and someone just tells you to build a cabin using whatever you can find. It's messy, you have to improvise, your tools might literally break in your hands, and there's no single right way to chop down a tree.
That's a really great way to visualize exactly the problem with prior bioinformatics benchmarks. Things like BixBench or BioAgentBench.
Yeah, those older tests.
Right, they were way too prescriptive. They practically handed the AI the IKEA instruction manual, telling it exactly which tools to use and what steps to take. And that severely limits our ability to test if the AI can autonomously formulate the scientific strategy.
It's just testing its ability to follow orders, not its ability to actually do science.
Exactly.
So how did the Genentech and Roche researchers actually solve this? Because they obviously needed a test that was completely objective and ungameable, but still required the AI to actually think like a scientist navigating that forest.
Well, they used a pretty brilliant strategy involving synthetic data, augmented data, and metadata scrambling.
Okay, that's a lot of jargon. Let's take those one by one.
Sure, let's break down synthetic data first. Imagine the researchers generating a completely fake genome using highly specific mathematical rules and then generating fake DNA reads from it.
So they're playing God in a tiny digital universe.
Basically, yeah. And because they made it, there is a 100% objective, ground truth answer. They literally created the universe it lives in. But the AI doesn't know the rules of that universe. It has to use real-world bioinformatics tools to discover that hidden answer.
It's like a scientific escape room. I love that. And what about the metadata scrambling? Because when I was reading the source material, I found this part incredibly clever.
Oh, it's totally ruthless.
Yeah.
So for metadata scrambling, they took real-world data sets, for example. They used an ATAC-seq data set from an axolotl.
Okay, quick pause for you listening. ATAC-seq is a technique used to look at chromatin accessibility. It's basically a way of seeing which parts of the DNA are open and actively being used in a cell. And an axolotl is, well, a really cute salamander that can regenerate its limbs.
Right, thank you for that. So they take this axolotl data set and they intentionally swap the tissue labels They basically just lied to the AI.
Wait, so they handed the AI a file labeled liver that was actually like heart tissue.
Exactly. The AI's task was to process all the raw data, look at the underlying biological signals, realize the labels didn't match the biological reality of those tissues, and then identify exactly which two tissues had been swapped.
Wow. Okay, so just to do a quick recap here to make sure we're all on the same page. The researchers created an environment where the AI absolutely cannot just memorize textbook answers. It has to navigate real noise, identify human errors or inconsistencies, and mathematically prove its work.
That's the benchmark in a nutshell.
But I do have to push back a little on how they actually administered this test, because from what I read, they dropped these agents into what they called a bare minimum compute environment.
Yes, the bare
Like, you're dropping a digital agent onto a blank, empty computer with just a text prompt. Isn't that just setting them up to fail? I mean, even human PhD scientists struggle for days just fighting with tool dependencies, broken Python packages, and outdated GitHub repositories.
That is precisely the point. Setting up the environment, finding the right tools, troubleshooting broken code that is half the battle in real computational biology. A real scientist doesn't just sit down at a perfectly configured supercomputer.
They have to build their own pipeline.
Right. And the baseline results from this bare minimum setup are the big reveal of the paper. When they tested non-agentic LLMs, meaning just the standard ChatGPT or Claude interfaces where you type a prompt and just get text back, they completely failed.
Like, how bad was the failure?
They scored around 3 to 5%.
Oh, wow. Which is basically just random guessing on multiple choice questions.
Exactly. Because they're just brains in a jar. They can't actually do anything. They can only talk about doing things.
Right. But the agentic systems, the ones equipped with the command line interface, the ability to think, browse the web, write code, execute it, read the error message, and try again, they absolutely crushed it.
They really did. Codex CLI, which is powered by OpenAI's GPT 5.4, hit 83.3% accuracy, and Claude Code Opus hit 81%.
That is a staggering jump, from 5% to over 80%, just by giving the AI the ability to interact with the environment.
It changes everything.
To kind of conceptualize how this works for you listening, think of the acronym ACT. Analyze, Code, Test. That is the agentic loop.
The ACT loop, yeah.
The AI analyzes the problem, it codes a solution or downloads a necessary tool, it tests that code, it reads the inevitable red error message, and then it leaps back to analyze what went wrong. It literally claws its way out of dead ends.
And to truly appreciate that 80+% accuracy, we really need to look at exactly what these agents were doing behind the scenes during those loops, because it frankly feels like reading a sci-fi novel. Let's look at the Saluki model case study.
Oh, this one absolutely blew my mind. So the task was in the machine learning domain. The AI had to optimize an mRNA sequence using a very specific published deep learning model called Saluki. That was it. That's the entire prompt. Optimize the sequence using Saluki.
Right, and the logistics that the Codex agent navigated to solve this were just insane. First, it searched GitHub and realized the Saluki model wasn't a standalone, easy-to-find repository.
It rarely is.
Exactly. It was hidden inside a different, much larger repository. So it cloned that repo, searched the underlying code, and found a secondary repository mentioned deep in a readme file.
It's literally playing detective.
It gets even better.
Yeah.
To actually run the model, it needed the pre-trained weights. Think of the weights as the actual brain or the memory of the deep learning model. Without them, the code is just an empty shell. It's useless.
So where were the weights?
It extracted a Zenodo DOI, a digital object identifier, from an image badge hidden in the Markdown file. Zenodo is this open access repository for research data. But when it finally navigated to the data, it realized the weights were trapped inside a massive 18.75 gigabyte archive file.
And let's be clear, if you're working in a bare minimum computational environment, trying to download 18 gigabytes just to extract one tiny file is a nightmare. It's going to crash your system, run out of storage, or at the very least just take forever.
Exactly. So what did the AI do? It used HTTP light range requests.
Which is so smart.
It essentially sent a highly specific network protocol message to the server saying, hey, do not send me the whole 18 gigabytes. I have calculated exactly where the file I need lives inside this massive archive. Just send me the exact 104 megabytes of weights located at this specific byte offset.
That is absolutely brilliant. An analogy for you listening. It's like walking into a massive multi-story library to find one specific quote in a 10,000-page encyclopedia. Instead of lugging the massive book all the way home, flipping through it, finding the quote, and then throwing the rest of the book away, the AI just takes a photocopy of page 404 and walks out.
That's the perfect way to put it. And then, as if that wasn't enough, it programmatically injected missing NVIDIA GPU libraries into its own Python environment just to make the code run faster. It finished the whole thing in 12 to 35 minutes. The researchers noted this would take a human expert 3 to 4 hours of frustrating troubleshooting.
It's an incredible display of engineering autonomy, but we also saw incredible displays of logical autonomy, right? Like the 1,000 genomes case study.
Yes. The agent had to cross-reference individuals in the African Functional Genomics resource with the 1,000 Genomes project.
Wait, cross-referencing two completely different global databases sounds like an absolute nightmare. I mean, human labs can't even agree on how to label a basic spreadsheet. Did the researchers have to hold the AI's hand to map everything? It's not at all. It autonomously scraped FTP sites, which are basically old-school, bare-bones file transfer servers, reading through raw HTML index pages just to find the exact demographic files. And to your point about naming conventions, the 1,000 Genomes project used the prefix NA for certain cell lines, while the other database used GM, and no one told the AI they were the same thing.
So how did it know?
It parsed the metadata, recognized the alias pattern entirely on its own, realized that NA12878 and GM12878 were the exact same biological sample, mapped the IDs perfectly, and then downloaded only the necessary intersection of data.
Okay, let's pause for a mini recap here, because this really makes you wonder. If a piece of software can autonomously track down its own tools, troubleshoot its own errors with complex NVIDIA drivers, and scrape raw FTP sites to resolve human naming errors, what exactly is the day-to-day job of an entry-level bioinformatician going to look like next year? That is the million-dollar question, isn't it? But before we get too carried away, we really have to look at the flip side. If these agents are performing superhuman optimization and data scraping, why didn't they score 100% on the benchmark? Where exactly are the edges of their intelligence? Because, to be clear, they do fail.
Right, and when they fail, it's rarely because they lack the raw compute power or the coding knowledge. It's because of what the researchers call the pitfall of plausibility.
The pitfall of plausibility is fascinating. It stems from how large language models are fundamentally trained from the ground up. They're essentially advanced prediction engines, designed to output the most statistically plausible next token or sentence.
Right, they just want to give you an answer that looks correct.
Exactly.
Yeah.
So when an agent is solving a complex problem, if it finds a solution that looks mathematically or biologically plausible, it just stops. It satisfies the prompt, it suffers from premature stopping, and it entirely misses the deeper, more complex biological nuance that a real human scientist would catch.
It's kind of like a brilliant student taking a really complex math test. They finish the front page in 10 minutes, look at their answers, feel incredibly confident because the math checks out, and they just turn the test and completely missing that there were 5 more incredibly difficult questions on the back of the page.
Yes.
They aren't fundamentally incapable of solving those questions. They're just dangerously overconfident in their initial success.
That's a great analogy. Let's look at how this plays out in this synthetic multi-mapping case study.
Oh, this one is deep.
It is. The agents had to estimate the proportion of reads coming from 4 synthetic chromosomes. Now, in genomics, multi-mapping reads are basically short snippets of DNA that are identical in multiple places across the genome.
So they could belong to a bunch of different spots.
Right. If you just do naive counting, meaning you just match a read to the very first place it fits, you get a completely wrong answer because you end up double or triple counting certain regions.
So how did the agents handle it?
Well, the agents that fell into the pitfall of plausibility just did the naive counting. It looked like a standard bioinformatics pipeline. It produced a very plausible-looking spreadsheet of numbers, so they just stopped and reported the wrong answer.
Confidently wrong.
Exactly. But the successful agents, mostly Codex, on this specific task, realized the trap. They recognized the multi-mapping issue and actually wrote a bespoke expectation maximization algorithm from scratch to solve it.
Let me jump in here because expectation maximization, or EM, is such a brilliant mathematical concept. For you listening, imagine you're a detective trying to figure out which of 2 suspects walked through a muddy room, but the footprints are super blurry and overlapping.
That's a tricky scene.
Yeah, you can't just count them. So you make an initial guess, that's the expectation about which prints belong to who. Then you use that guess to refine your model of their shoe size, which is the maximization step. Then you go back and reassign the footprints based on your new, better shoe model.
And you just keep doing that.
Exactly. You repeat this loop over and over until your theory stops changing. The AI realized it couldn't just count the blurry DNA reads. It recognized it needed this advanced statistical loop, and it just built it.
It's a profound display of reasoning. But then, on the other hand, we have cases where the AI's extreme confidence leads it straight into a brick wall, like in the transcription factor overexpression task.
Okay, set this one up for us.
So they had to find which protein was overexpressed using ATAC-seq data. Claude actually succeeded here because it relied on a standard, highly trusted bioinformatics tool called Homer. Codex, however, failed completely.
Why did it fail? It just built an EM algorithm from scratch.
Because Codex confidently decided it didn't need pre-built tools. It wrote its own custom analytical pipeline from scratch.
And it forgot a crucial step. It forgot to match the GC background sequences. Let me explain why this is a fatal error, just to be thorough. DNA is made of 4 bases, A, C, T, and G. If you are looking for a specific pattern, say a string of DNA that is mostly G's and C's, you absolutely need to know the baseline rate of G's and C's in that specific part of the genome.
It's the control group, basically.
Right. If you don't control for that background rate, you'll get massive false positives everywhere. Codex completely missed this fundamental biological control step. It built a beautiful, highly efficient, but scientifically flawed pipeline, and it just confidently failed the test.
It really makes you think about how these different models process information. And that brings us to a really interesting insight from the paper. Codex and Claude actually have very different brains based on how they were trained. And they approach the exact same problem in radically different ways.
I love this part of the deep dive, the 40 nucleome case study.
Yes. So the task here required the AI to find a differential chromatin loop between 2 cell lines using Hi-C data. For some context, Hi-C data is an incredibly complex data type that measures how the 3D genome folds inside the nucleus.
Right. Imagine the genome isn't just a straight line, but a giant tangled ball of yarn. Hi-C data essentially tells you which pieces of yarn are touching each other inside that 3-dimensional ball. Finding a specific differential loop is like finding a specific unique knot in that yarn that exists in one cell but doesn't exist in another. So how did Codex do it?
Codex acted remarkably like a human bioinformatician would. A human would go to a web browser, pull up a visual mapping tool, and look for the loop in a specific region. Codex used web APIs to pull localized image tiles from the database, essentially querying only the specific region of the map it cared about. And it found the answer in about 7 to 22 minutes. It was totally surgical.
So elegant. It only asked for exactly what it needed. And what about Claude?
Claude went full brute force. Because Claude Opus is trained for massive context synthesis, it basically wants to read everything. Its instinct was to ingest all the data. Instead of using the visual API, it tried to download 24 gigabytes of raw, multi-resolution contact matrices.
Oh, no.
Yeah. It wanted to computationally recall all the loops for the entire genome from scratch. It got completely bogged down compiling C++ libraries to process the massive files, and it took almost an hour.
It's the classic work harder, not smarter trap. But this leads right into the cost and performance breakdown of these models. How practical is it to use these agents right now, given these completely different approaches?
Well, the stats from the researchers are quite revealing. Codex, running GPT 5.4, is generally faster, averaging about 11 minutes a task, and it's much cheaper because it's so efficient. Claude Opus 4.6 is slower, averaging 18 minutes, and it is the most expensive model tested, coming in at $1.70 per question.
$1.70 for one question. I mean, that adds up incredibly fast if you were processing hundreds of thousands of variations in a whole genome.
It absolutely does. However, there is a catch here. On the absolute hardest questions in the benchmark, the ones labeled levels 4 and 5, which require massive leaps of logic, Claude Opus actually beat Codex. It scored 69% compared to Codex's 59%.
Wow, really?
Yeah. So Claude's brute force, methodical, download everything and read it, nature actually pays off when the biological logic gets incredibly convoluted.
Okay, so what does this all mean? If you, listening to this right now, want to use these tools in your own research or work tomorrow, what is the ultimate takeaway? Let's bring it all together.
The big takeaway from the Agentic and Roche researchers is that general-purpose AI systems are absolutely no longer just conversational chatbots. They are highly capable, autonomous agents. They can wrangle data, write bespoke scripts, and discover complex tools on the internet without your help. But because of their brittleness, that pitfall of plausibility we talked about, you cannot just set them and forget them. The researchers strongly recommend breaking large scientific tasks into 30 to 60-minute chunks. You really need a human in the loop to audit the intermediate steps, check the biological controls, and make sure the AI hasn't confidently run down a scientific dead end.
Right. You're basically a manager now, and the AI is your brilliant, tireless, but maybe slightly overconfident new intern. As these models continue to scale up in parameter size and capability, testing them on strict, objective benchmarks like CompBioBench is really the only way we will build actual trust in them as autonomous scientific co-pilots.
Exactly. We have to ruthlessly map their limit and deeply understand their blind spots before we can blindly rely on their discoveries.
And that leaves us with a final, provocative thought for you to ponder. We've seen today that an AI can already independently navigate undocumented code bases. It can selectively download fragments of massive data sets using advanced server requests just to save time. It can write bespoke expectation maximization algorithms from scratch just to pass a test.
It's doing the heavy lifting.
So, what happens when we stop testing them on synthetic data where we already know the answers? What happens when we finally take off the guardrails, point these autonomous agents at the noisy, uncharted, entirely real data of a human genome, and simply ask them to find the cure we've completely missed?
That is the frontier we are standing on right now.
Thanks for taking the deep dive with us. Until next time.